Now for the final talk before lunch, it's Patrik Sjöberg and Martin Bekken. Patrik is a former high jumper with several medals from Olympic Games and World Championships, and Martin Bekken is works for a company called BEVI, where he's CSO. I guess it means Chief Scientific Officer. Chief Sales Officer. Almost the same. Okay. Please, Patrik and Martin.
Thank you.
Ska jag styra?
Du får stå vad du vill.
Okay.
Okay.
So, I'm going to help these guys out. Really powerful speeches so far. You can see that these two are not scientists, so you will get a different
Can be tonight.
point of view from them. So I'm going to hand it over to Martin to explain a little bit more about BEVI.
You can take the next one, Team BEVI. Yes, we are a company from Norway who is growing with the salmon industry, and we have growing a lot the last 10 years. So today, last week, I think we have a turnover of 500 million euro, and it's difficult to grow fast. And then I find out 3-4 years ago that's a good way to live with the value, core value I have a company is to do something instead of just giving money. So we start Team BEVI, and I met Patrik. So we start bicycling around Nordic countries to save to foundation money for different things for kids. That's right, Patrik?
Yes. I have a background as a high jumper, and normally we don't run, we don't bicycle, we just jump. So when I met Martin, he had a crazy idea that we should bicycle from Dorotea. It's a small town up in north, and Swedish people, they think north is above Stockholm. So one night we had a little bit too much to drink, and he said, let's go from Dorotea to the main office in Norrtälje. And it sounded okay until we look at the map. It was like 500 kilometers.
750.
750 kilometers. So we did a race, the first race from the main office to Vårgårda, and it's just across Sweden. On the map it looks like a pleasant ride. But it was 600 kilometers in 3 days.
Yeah, and Patrik have never bicycled before.
So we had a lot of fun, and we raised a lot of money that we could give to the Cancer Foundation here in Sweden. So I think the company, they do it because it's meaningful, and I like to do things that are meaningful. And I think a lot of famous people, they just do things to get more media, and they never think about what they actually do. And I think this is, it's perfect, even though I hate bicycling.
Yeah, and me.
Yeah, and you.
Everybody knows that Patrik still have the European record. That's right?
Yes, in high jump.
242.
Yes.
But he is not best in Sweden. Yes, he is. Okay.
But you have a different history also from your career. So helping out from other companies and stuff like that, not just BEVI.
Yes, 2011 I came out with my second book, and I explained my background as an athlete. I was never seen as a real athlete because I was smoking and drinking, and I had a lot of bad media.
You had a lot of fun.
Yeah, I had a lot of fun. But in this book, I explained that I got sexually abused. My coach for 3-4 years when I was 10 to 14. And that explained why I did a lot of crazy things. So the same day I came out with a book, I started to work with these questions, because we have a terrible statistic in Sweden and in Europe and in the whole world. We actually have, in average, 3 kids in every school class get sexually abused in Sweden, and the statistic is the same all over the world. So A big part of my work today is to work for this foundation, 3 ska bli 0. It means that 3 shall be 0 in each class. And every time we do bicycling now, we divide the money for this foundation and for the Cancer Foundation.
For Tuvas Dag. Yes, BV Group, we are a company from Norway, I said it before, and we are now in 8 countries. And Patrik and me, we're going to try to bicycle to every plant we have. And so today, I think we're going to agree tonight to bicycle from Denmark to Netherlands. So if somebody want to join us, you can sign tonight. Roger, you're okay. Yes, we go
but
next.
a lot of the core values is something you found out 4 years ago.
Yeah.
And that's...
It's I'd say it now. We have people. Was some of the speaker here before? Was you? It's so much business in the world right now. I'm living in the company. We're talking money. Every day is business. You buy new company, you sell a new company, it's money. But 4 years ago, when I started I found out we need to, instead of talk and give money, we need to do something. So we start a lot. We have our core values on this stone. So I start this with Patrik, and we start take care about immigrants, and we start many different things on the social, and then have giving so much for our company and for the customers, for the people who works and everything. We are 35 different countries, people from countries who work in our company, and so we like immigrants. Without immigrants, we cannot live in Sweden with our company. So this is what we have done, me and Patrik. And me and Patrik and many more. I think we have around what is it 800,000 Swedish crowns, 700,000 Swedish crowns we have given away to different things. And
there is actually a fun note that this morning you had a big drop-off at the Cancer Hospital for Kids.
Yeah. And this is, he's gonna explain a little more. The founder of Tuvas Dag is here. Tuvas parents is not here, but the founder is here. So he's gonna come up here on the stage now. Last weekend, I think we can take the next. You can put on the movie.
Then I need your help.
Yeah. Last weekend we have bicycling from Gothenburg to Varberg. Patrik lives in Gothenburg and I live in Varberg. So we have a race, so we're calling 242 Charity, and we was around 100 bicycle to Gothenburg to Varberg. And there is Patrik Ekvall, is a good friend too. There is Mr. Sjöberg. And this day we was money for Tuvas Dag and 3 ska bli 0.
Yes.
This kind of bicycling we do around Nordic countries, and now we're going down to Netherlands too.
It's no mountains in the Netherlands, no?
No, but in Norway it's mountains.
Yeah, in Norway.
Yeah, we always say that. I promise always, Patrik, 100. What did we say? 100 kilometer maximum.
Yes.
And then I saying I showing a map, but I always showing wrong map.
Yeah.
So it's 150 kilometer. And then we start. And then we have
no hills and no mountains.
No. But I can explain a story when you see this. We was in Denmark, and
yes.
I promise, Patrik, it's no mountains in Denmark. And the first we start was going up in 30 minutes for like...
Yeah, to the highest point in Denmark.
With no mountains.
Yeah.
But
this is Team BV. But Team BV is, this time it's Tuvas Dag and 3 ska bli 0. And Tuvas Dag is something the founder is there, is Martin Larsson. He started Tuvas Dag and Varberg Fleet 3-4 years ago. Stand up, Martin. And this man, he's doing this without you don't take any money for that work you do. You have, we can show later. We can start a movie about Tuva. Tuva is passed away 2 years ago. Yeah. And her parents is not here today, but they normally are on this kind. Yeah. So this is the Tuvas Day.
So.
Yes. So Martin, you can explain how many kids have you done? Stand up and talk now, okay? Is it 300? Or?
A bit over 300 kids in the 2 years.
And what are you doing with it? You?
We do individual prizes. Kids, they ask us what they want and we give it to them.
And we go to different hospitals and give away gifts. So today, this morning, we went to Lund and gave away gifts for about 40,000 Swedish crowns. So that's what we do. Pink Santa during Christmas, you see up there. We go around 5 different hospitals this year. So we try to give the kids and the families and the brothers and sisters something to remember. And it's all, we have no fees at all. We do this for free. So thanks to Bevi and Patrik, we can do this, and other companies.
And you have some gifts with you here.
Yeah.
You can come here. And the three, vad heter de, cheferna?
Det är ju Georgi. We have Roger and Johan.
So now you are in the team. So next bicycling, you are.
So what I have here
Do I get a bike?
Yeah.
It's a shirt that we have, because everything we do is in pink. We have pink cars and pink shirts, and everything is in pink.
Pink underwear.
Yeah, I got it.
And you need to put it on so we can take a picture.
Very well.
Okay.
One size fits all.
Stretch.
So the thing with Tuva, the movie you saw was the first day we did a surprise. So after that we made Tuva's Day. And so last year I made this tattoo. So every day when I have a little bit of problem with energy, I just look at that and then it just go.
And when Patrik bicycle, he have a lot of problem with energy. We take a picture with him, Patrik. Come on. Point in. Okay? Yeah? Okay? Okay. Okay. Ni ser lite atletiska ut. Ja, nu kommer den. Okej, är du ready? Okej? Okej. 1, 2, 3. Yay!
Alla har inte tränat än.
The first presentation this afternoon will be given by Jessica and Peter Ekolind from Ängelholm. Ängelholm, for those of you who don't know, is in the northwestern part of Region Skåne. It's a beautiful small city there. So please, Jessica and Peter.
Well, thank you very much for inviting us to this symposium today. And we will try to do this interactively. So when you see my not blue shirt, it's blue pictures, and you see my wife's red, it's red pictures. So you don't
Very easy.
get
so.
lost. Okay then. So our cancer experience started in 2014. And in the beginning, I was quite physically active, 2 hours running, exercise at gym a day, and felt very well with that. And no smoking, healthy food, no hazards in my work or at home. So I wasn't supposed to be afraid of anything. But I felt nocturnal pain in my right shoulder, and an MR and CT showed a tumor, approximately 7 centimeters, located here in the breast. And a follow-up biopsy showed a very aggressive T-cells lymphoma, so I got cancer. It was like somebody just put on me and my family's life bottom on hold or pause, like that, from one day to another. So how to plan for further life then?
So what happens then in the head of the relative one? I'm standing here and you have heard some other stories before us. You can see this as a very busy slide, but this is actually my life, and this happened in my head. I was thinking about my job. I felt alone. It was sorrow. It was sleeping problems. A lot of questions, but no answers. Where should I park when I'm visiting the hospital? It was big questions and smaller questions, but all in all, I was very affected, and my family as well. And we have 4 children together, so yeah.
So back to treatment then. And for this kind of cancer, the therapy choice number 1 is chemo. So my oncologist said, let's go for the golden standard. It's a combination of different chemotherapy drugs, and we call it CHOEP. And it works good for many cancers. And I was in good shape, trained a lot and healthy and everything else. So I said, let's go for the worst. I can take it. Just get rid of it and do it fast, please. So I was admitted to the oncology department here in Lund in the 1st of April for my first treatment. And this was a treatment for every fortnight in 6 courses. So I can say that end of June, when I finished it up, I was not so tough anymore, because it harms your body quite hard. All the hair was gone, blood levels were low, and I felt tired, and I was quite unfocused on work. It was difficult to keep up the focus and the concentration. But end of summer, a PT scan showed no cancer residue, so this was good. And then I end up in the follow-up period with the CT scans, blood samples, and doctor's visits. But they, after time, come less and less frequent, and no signs of any relapse. So 2 years later, in March 2016, my oncologist said, we don't need to follow up you anymore. Your kind of cancer is very aggressive, and if it hasn't showed up in 6 months, that's good. So we can stop here. So you are now cured from this cancer. And I have to ask him to repeat that 3 times before I understood or could take it in. So family and friends celebrate. We went to Bali to have a nice time. And life was good again, and the play button was on. Perfect, isn't it? Or was it really so? I wished that we could have stopped here, because then life is gone. But just a few months later, during the summer, I felt a little bit tired. Running was harder and harder. I skipped the gym one day after another. I didn't have the energy to go up and exercise. But nothing dramatically, so I thought this might be a mental reaction following the cancer and all that stuff. But this will be better after the vacation period. You know, you come back in routines again, and friends sitting late at night at home and so on. But on September the 1st, I was acute admitted to the hematology department here in Lund again, and isolated. I had very low red blood counts, negligible levels of platelets, and practically no white blood cells left. I got a new cancer, an acute myeloid leukemia and AML. And this was, how was this possible? I mean, I was cured 3 months before, and now I was sick, and this time I was really sick. And now the pause button was huge. I mean, the first cancer we survived, moved forward, but now it was completely stopped. The prognosis is very poor on this kind of cancer compared to the first one. And from one day or from 1 hour to another, it was stopped again.
So some reflections from me as a relative after 2 different cancers. As being the person always standing beside, I think this first sentences describes this quite good. The relative perspective is almost always seen in a relation to the patient perspective, and the related becomes someone whose needs are met by the patient needs. I don't know if anyone really understands this, because of course I would like the healthcare to focus on my husband being well and being treated from his cancer, but no one sees me, and I think that this is a problem. Yes, and relatives can have difficulty formulating their own needs because they do not want to take place from the patient, as I said. So I felt very much alone, and it was not. I was not very much asked either from the healthcare professionals how I felt. I was someone in the room but not seen. Yeah. The treatment of close relatives as a separate individual with their own needs is important, needing a separate place in care. And I really do believe that we can help each other. We are a big group now of relatives that could really help each other in this, I think. It's just about organizing.
So the next cancer, next chemo treatment started over a day. And I had got an extreme high-risk therapy-related leukemia. And investigations afterwards showed that this was caused by the chemo I received for the first cancer in 2014. And you can say that shit happens, because it does. But we felt that this were very unfair and unnecessary, actually. Because I was cured and that now I was in really bad condition. So 4 new courses of chemos, but in this time I would say it was with my life at stake. And during this chemo, it goes to the bone marrow because the cancer is there. It almost killed the bone marrow. And if you do that, you don't have a working immune system anymore. So during those neutropenic episodes, I received 2 septicemias, and one caused by a multi-resistant bacteria. And I was really ill. You were spending all your time at the hospital at that time.
Yeah.
So the doctors have to take some decisions for life, I would say. And very soon, they start looking for a donor for a stem cell transplantation. Without the stem cell transplantation, with only chemo for this kind of leukemia, the 5-year survival is about 10%. So something has to be done. And I was lucky. A match was found with a 20-year-old male from Germany, and on January 19, I was transplanted with his stem cells. And 3 weeks later, I was back home. In total, 3 months of hospitalization and isolation from family finally comes to an end. A very long recovery period started, but after the last summer, I felt relatively good shape again.
So what did I do meanwhile? I tried to get support wherever I could from friends, from other cancer patients and relatives. But then a colleague told me about this new course at the university here in Lund. And it's called Patient and Self-Care Ability. So this was a course for patients, for relatives, and also for cancer patients. And we were a group of 30 people. It was a distance course, so we only met 2 times in Lund physically. But this was a really, really good way of handling what I have been through. And actually, it also showed that we can cooperate together, patients, healthcare professionals, and the relatives. It was shown in this course. So this gave me a lot of help, strength, and hope as well, I would say. So what is then important for maintain health as a relative? To be given a possibility of inclusion in healthcare, to be included, I think this is very important, because if you're always the one standing beside, you get even more worried. And when you get more worried, you don't sleep. And when you don't sleep, you cannot work. And you have to take sleeping pills and so on. So this is a negative trend. See the related as a resource. I have a background in the healthcare, so of course I am a little bit equipped, so to say. But I would say that we can help each other, use the related ones as resources. Counseling is needed to maintain some form of balance in life. If healthcare is insufficient, often other close relatives can help. I'm thinking in terms of a cancer coach. We can help each other. And this was also something we discussed during this course that I attended to. So I think it's possible. And of course, my wise husband's mantra during his 2 cancers also applies to close relative ones. Eat, sleep, drink, and try to exercise. I mean, I don't know how many times we have been walking around this building. Yeah, a lot. And I think this is doable. You just have to push yourself and perhaps help others to push themselves a little bit.
So I think the reason for we are here today is a good initiative, and it's needed good initiative, because we see the cancer development and so on. But still there is some unmet medical needs. And we need to, for sure, have better tools for predictions of dangerous, serious adverse events, like mine. Actually, my 2nd cancer is an adverse event. There is also a need for much more specific treatment that not injures the healthy organism, like old chemos does. And seriously, is a derivative from a chemical weapons that used in the Second World War still the drug of choice in 2018? Yes, it is. And the C in the combination Schar is cyclophosphamide. That's a chemically related to nitrogen mustard. It was used as a chemical weapon in the Second World War II. That's actually how they found out that this was very aggressively on the bone marrow system. And it was registered as a pharmaceutical in Sweden in 1959. The second one in the combination is etoposide, and that's from 1986. So a question is that how many patients should accept that the standard treatment for the cancer could give them a therapy-related and even worse cancer? I appreciate that I wouldn't been here today if I haven't got the Schar combination in the beginning. But still, do we need some more efficacious and not so aggressive treatments? So I think that the risk ratio on a group level is maybe acceptable, because you actually find out that a few percentage got this late reaction, harmful effects. But on the individual level, if it's not acceptable. Absolutely not, I can tell you.
So, thank you. The targeted team, health professionals, patient and relatives. What I found out is important is to have an early meeting with a contact nurse who can be a help for the whole family, not only the patient, and pilot on other resources that may be needed during the treatment. It could be cancer rehabilitation in a very, very early stage. This comes in more or less, perhaps a little bit too late. But I think this is one part that I think is very important. Customize a conversation. A doctor who listens and provides the opportunity for the related parties' questions. Sometimes these meetings are very quick, effective, giving the diagnosis. You have time for some questions, but I miss perhaps a more adapted kind of meeting with a doctor that is, again, done for the specific patient and the relative. Yes, and individualized. Someone here in the audience also brought this up in her lecture. individualize the care plan. You should individualize the treatment, but I think individualize the care plan so that you have something to follow for both the relative and the patients. Because if you don't have something to hang into, you worried a little bit extra, I think. And if you do that, you're in this bad circle again. Yeah, and I've mentioned this before. A given meeting place for relatives already at the hospitalization. It could be like a relative coffee corner. It could be, I don't know, some small room to have like meetings with other relatives in the same situation as yourself.
So, finally, from us, many thanks for letting us give you our story with you today. This is our story. It looks very different from patient to patient, but on the other hand, you can pick up some common themes. And I will not say that I'm cured. I don't dare to do that anymore. However, if you're wondering how my life is and our life is, it slowly comes to play again. Thank you very much.
Thank you.
Thank you very much. Now it's time for a person from the category of collaborators that we work together with, and this is Thomas Conrads. He is Associate Director of Scientific Technologies in the Inova Schar Cancer Institute in Virginia. Please, Thomas.
Thank you, Jon, and Georgie, and Peter. Thank you very much for having me. Hats off to you, Georgie. This is an amazing day. It's even extremely hard to follow these things with science. Absolutely. So I'm going to take you on a little bit of a recent archaeologic dig and describe to you, I think, some of the important milestones that have got us here to even be able to talk about proteogenomics, to be able to initiate moonshot type of programs. And then I'll tell you a little vignette about how we've seen some success using tissue-based proteomics in an extremely dreadful cancer. So the archaeologic dig for me begins about 65 years ago with these two gentlemen who, oops, I'm going to do this now. With the benefit of seeing Dr. Franklin's picture, we're able to build a model of what turns out to be the double helix of DNA. This served a very important purpose because what it did is it allowed us to start to nail down one of the central tenets of molecular biology, which is the central dogma, which all of us in undergraduate and graduate school in biology learn about, which ultimately relates to the continuum of DNA transcribing RNA translating proteins. And what the structure of DNA did is it allowed us to understand how DNA replicated itself. Seems like a very basic tenet, but in fact that started the molecular biology revolution. And it was a few years later, in 2001, when I actually, a year after I started my professional career at the National Cancer Institute, that the announcement was made as to ultimately the solution of, or the description of the human genome. Actually, it was Craig Venter's genome that was described first in 2001. Here's the press release from Bill Clinton. And what this all promised was the following. It promised essentially the ushering in of a new age of medicine. We expected from this genomics revolution that we'd be able to identify risk factors, identify prognostic and diagnostic biomarkers, and develop new targeted mechanism-based medicines. That's what we expected, and that's what we fully anticipated in 2001 would happen from this monumental discovery. And so about 3 years later, the National Cancer Institute launched a program. It was a major program funded with over $600 million to ultimately sequence multiple organ site malignancies. Ultimately, they accomplished this in 33 different organ sites. They generated lots of data. 2.5 petabytes. I don't know what that is, but I think David Fenio will tell us something about big data later. TCGA findings have allowed us to do a lot of things, which is provide tumor subtype classifications, highlighted alterations that may, and I put that in italics, may be actionable. Again, pretty expensive project, but we hoped that that would solve a lot of our problems in cancer biology and cancer treatment. I would say the culmination of the TCGA was ultimately this trial, which is called the MATCH trial in the United States. Again, this is a major effort to ultimately use the information from genomics to inform how to match patients best to the best drug based on a mutational profile of their tumor. So patients take their diagnostic tumor, they send it to one of 4 sequencing centers in the United States, a report is given back to the doctor, there's 24 treatment arms against which they may match based on a mutational profile. Currently, 6,000 patients have been enrolled, 4,700 actually had tumors, 722 were matched successfully. 15% were matched based on the genomic sequence that we have from these 6,000 patients and a $600 million program. So the question is, I think that's a great accomplishment. Infrastructurally, it showed that we could do this. We could take molecular information, generate a report, match it to something possibly that could be informative, and wind that exercise away. But I think we all agree in this room that we need to do better, particularly after seeing some of the very heavy testimonials we've seen today. We have to do better. And that, of course, is why we're all here. So the limitations, why can't we do better with this information? I think it's captured in these limitations that I summarize here from TCGA. If I go back, only primary tumor tissue was used. So no metastatic sites were sequenced in TCGA. Only those which were relatively pure. This is a sort of a wrinkle, but it turns out that impure tumors, that is, tumors that have more stromal infiltrate, more background tissue, typically portend a worse outcome. So they actually selected for better outcome tumors. Patients with prior chemotherapy were excluded, so we don't have any understanding of the impact of chemotherapeutic treatment of tumors. So we don't know how tumors might adapt, evolve. We know some of this from PET, sort of smaller boutique studies, but in this major national scale, we sort of failed at that. Limited clinical data, and no biospecimens from surveillance were available, so we couldn't watch the cancer patient's journey throughout these different treatments from that data. So where's the opportunity? I think the opportunity is in what we're here talking about today and what links groups like Professor Marko-Varga's and mine in trying to do international science, which is to try to build out this continuum and understand the complexity of what turns out to be a horribly complex system. So if we just simply focus on the genes and the mutations that the genes may hold, or the epigenetic alterations of the genome, we're getting some view of the picture, but in fact we're missing a lot of it. So this is really what the view of molecular biology looks like in a flat file. That is, every gene can be transcribed and spliced into probably 10 to 20 different forms. Those 10 to 20 different forms make 10 to 20 different proteins. Those 10 to 20 different proteins can ultimately be modified by 300 different covalent modifications, which can change their activity, change their localization. So now we turn from 22,000 genes or so to probably millions of proteins. So that's a problem if we only look at the genome. And so you all here are very familiar with proteomics. Here, I've been using this slide for 15 years and trying to encourage people to do proteomics. Ultimately, the point here is that genomic measurements do not reflect protein-level measurements. Things like protein function, localization, stability are completely lost with genomic information only. And in fact, it's even more complex than that because the proteome is highly interactive, so a drug for patient that may work for patient A may not work at all for patient B because the signaling may be going through a different network. And so we really need to tailor our understanding of the proteome to every patient. And in fact, it's more complicated even than that. Sometimes people call me Professor Complicated. There's 47 trillion of these cells in your body, more cells than we know about the stars. Okay? So And in each one of these cells, those proteins can be localized in different areas based on the modifications and have different activities. So it's horribly complicated. But I think we can still make progress, and it's sort of summarized here. Genotype does not equal phenotype, because this chicken has the same genome. Its really developmental program is driven by the proteome. So this has ushered in a number of initiatives to try to redress these challenges and fill in some of the gaps. So that's really the motivation behind the US Cancer Moonshot, which was announced January 12th, I know it well, 2016, by President Obama and taken over by Biden. Cancer centers, 3 of these cancer centers of excellence within the Department of Defense were called out. I co-lead the Gynecologic Cancer Center of Excellence, and we're playing a major role in the Apollo Moonshot. Specifically, I think in a good way, we have changed the dialogue from a genomic-centric dialogue to now a proteogenomic-centric dialogue. In fact, I penned this article that night after Obama talked, and it was one of the fastest acceptances in one of the highest cancer journals I've ever seen. So I think we're ready to embrace proteomics from a clinical cancer perspective. And so the Cancer Moonshot, one of the programs that you've heard about, Dr. Baker talked about, was this Apollo program. And this represents really a 3-way federal agreement between the Department of Defense, the Veterans Administration, and the National Cancer Institute to do proteogenomics to fill in the gap. And that's what we're trying to do. This is the Apollo emblem, and we're very active now. We're in full production mode for Apollo. The goal is to ultimately proteogenomic sequence 8,000 cancer patients newly enrolled into Apollo and follow these cancer patients and do serial surveillance of these patients. We have working groups, we have sub-projects in Apollo, and all of this is now in full production for about the last year or so. We've now completed Apollo 1, we've completed Apollo 3, Apollo 4, and Apollo 2 are being sequenced and analyzed by mass spectrometry currently. So stay tuned with that. I think there was a great quote Mark Baker had this morning from Steve Jobs, which is technology is really driving innovation, right? So this is, it's a little dark, but this was in 1999 or so, Mikhail Gorshkov and Dick Smith's group in Pacific Northwest National Laboratories with a 12 Tesla FTICR mass spectrometer. That was the state of the art in 1999, and that's what I cut my teeth on in growing up in mass spectrometry. Now it looks like this, right? And I can tell you the technologies, the throughput, the depth of coverage, the quantitative accuracy that we have in this little benchtop instrument far exceeds the $7 to $10 million instrument that we built in Dick Smith's lab. So we can dig very deeply into the proteome. We've been using proteomics for a long time in tissue. This was actually how I met Professor Marko-Varga, was our shared interest in tissue proteomics, and we've been doing this for well over a decade and a half and published many different papers. And what I want to do is I want to give you a vignette of how we've successfully used tissue-based proteomics in a really aggressive and hard-to-solve problem, which is ultimately esophageal adenocarcinoma. Here's a statistic for you. 600% increase in throat cancer in the past 3 to 4 decades in the United States. 600%. So those curves that you saw, that's sort of annualized all malignancies. But if you look at esophageal cancer, it's exponentially growing in the United States, probably related to the obesity epidemic, and I'll tell you why. So like many cancers, it goes through a developmental program, sort of like that egg to chicken, where normal lining of the esophagus goes through remodeling, and ultimately you can develop invasive carcinoma. There's a risk factor for esophageal carcinoma, and it's this precursor lesion, which is known as Barrett's esophagus. So here's some statistics. So it's not just increasing, but it's one of the most deadly cancers. And what do I mean by that? I mean the incident rate equals the mortality rate. So 5-year survival is only about 15% for this cancer, despite all of the chemotherapies, all of the surgical interventions that we've developed. Risk factors are rather generic. If you look around, a lot of us are at risk for this. What do we do when somebody has Barrett's esophagus? Well, what clinicians do is they So Barrett's esophagus, as I said, is this reorganization of the esophagus and metaplastic reorganization. It's present in about 10 to 15% who have long-term acid reflux. Patients with Barrett's esophagus are 150-fold increased in their risk of developing esophageal. So that's a risk factor. So we can put these patients on monitoring programs to try to detect cancer earlier, right? We heard about detecting cancer earlier. Well, here's your risk factor. And so what physicians do is if you're diagnosed with Barrett's, you go in every 6 months or a year, you get one of these things stuffed down your gullet, and every 2 centimeters up your esophagus, they take 4 biopsies. And they do that every 6 months to 1 year. So there's all sorts of problems with that because it's very easy to miss the cancer. It's very painful. That's not a very good risk monitoring program. So we decided to attack that, and I partnered with an esophageal surgeon, and we started doing tissue-based proteomics on these esophagectomies that he did to try to cure patients. So this is a total esophagectomy of a stage 1 esophageal cancer that Blair Jobe would do routinely in his clinic. And so we had the opportunity to go back into the pathology archives when I was at the University of Pittsburgh and do a retrospective analysis of tissues that had been collected over the years from Barrett's esophagus, from high-grade dysplasia, and from esophageal adenocarcinoma, to try to understand what the proteomic developmental program was to try to better predict who might progress. And so it's not that easy. We can't just take a tissue and make a smoothie out of it and expect that we understand something about the cancer, because cancer tumor microenvironments are actually very complicated. Here's just a sort of a cartoon version of the complexity of the types of cells that make up that tumor. And these are all vary depending on the tumor type, your tumor, the stage, the invasiveness, all of this. And so the amount of tumor cells and the background cells can vary quite a lot. So we just can't take that, mix up all of that noise, and expect to be able to pinpoint what's going on in the cancer. So many years ago, we started using a technique called laser microdissection, where we could use a specialized slide, put the tissue on that slide, impinge that with a laser, and do this under microscopic conditions, and collect cells specifically from the tumor microenvironment. So this is sort of hard to see, but this is sort of what it looks like. So we're really dealing with very, very microscopic samples. And ultimately, that's what we can do. We can collect specifically the cancer glands or the cancer cells away from the background stroma. So we can hone in on the proteomic signal from the cancer, or the proteomic signal from the stroma, or from the TILs, or, right? So we can very specifically and sensitively look at different compartments in the tumor microenvironment. So we did that for the case set that I just described. And you can see here, this is a hierarchical cluster analysis of the proteomic data that we collected. You can see here Barrett's esophagus, high-grade dysplasia, and adenocarcinoma. And even my 7-year-old can see that these pretty distinctly cluster from one another. So we selected the top 5 differentially expressed proteins that most strongly sort of associated with each one of these 3 classes. And then we went back and said, well, we don't want to just basically redo what pathologists are doing and look at biopsies. We want to try and see if we can see these in the blood. And so we identified from those 5 proteins that we could use ELISA assays, which are antibody-based assays that you can use on blood-based proteins. And so we did that. We developed ELISAs for those 5 proteins, and we collected serum sets from independent patients. So we collected a discovery set of about 60 or 70 serum samples from patients with gastroesophageal reflux disease compared to esophageal adenocarcinoma. And we tried to validate these 5 proteins. Doesn't matter what they are. And then we took those, and we got an independent serum sample set of about 100 other patients, tested these in those patients, and identified that, in fact, 4 of those validated pretty well. 1 of them didn't, but 1 out of 5 isn't bad. So we then came up with the opportunity to develop a diagnostic algorithm to say how well, based on the abundance trends of these proteins in these 2 high-risk groups, right, could we then diagnose whether or not you had esophageal adenocarcinoma in the background of the pathology of gastroesophageal reflux. And then we tested that using different statistical techniques, and we came up with a diagnostic accuracy of 93%. And so this represents sort of the paradigm that we use, and this is very similar to what Professor Marko-Varga is doing, where we start with the tissue, we start where the pathology is, where the cancer bioprocesses are happening, and then we aim to move that out into a deployable assay that is something much more sensitive, much more amenable to sort of monitoring risk in these patient sets. So with that, just like to acknowledge the huge group of folks that are involved, and definitely acknowledge our newfound collaboration in melanoma and doing very similar things with the melanoma environment with Lund University and Professor Marko-Varga's group. And finally, I do this in all of my talks. A special thanks to the patients and their family members for partnering and committing to this. So we biobank very actively in our group, and this is something up in our clinic because we asked one of our patients, tell us about your journey and give us some advice. And so she penned out that, which is, this may be a normal day at work for you. It's a big day in my life. The look on your face and the tone in your voice can change my entire view of the world. Remember, I'm not usually this needy or scared. I'm here because I trust you. Help me stay confident. I may look like I'm out of it, but I can hear your conversations. I'm not used to being naked around strangers. Keep that in mind. I'm impatient because I want to get the heck, she was being nice, get the hell out of here. Nothing personal. I don't speak your language. You're not going to do what to my what. I'm only here for 4 days, but I'll remember you the rest of my life. Thank you.
Thank you very much, Tom. We'll go on with the next presentation, which is given by Chris Allen, who is a molecular biologist at ThermoFisher.
Yes. Next, back. Okay, is this a laser pointer? Thank you. So, first of all, thank you for the invitation to come speak today. It was mentioned earlier in the day about bravery, and what struck me today, I'm actually attending quite a number of different cancer conferences. This is rather unique experience, and the bravery that's been shown by the patients, families and friends today, I'd like to acknowledge. So, you know, Eve, Mary, Peter, Jessica, all those people, Agnes, sorry. All those people, I'd just like to acknowledge that they were so brave to stand up here and give us their story. So thank you for that. So I'm going to focus on immunotherapy, particularly with regards to cell therapies. So this really has, certainly from my perspective, on the technology side, there's been a real explosion in these types of therapies moving forward. Essentially, there's a few different types of approach for immunotherapies, but it's something that's really become apparent in all the pathology labs because, of course, before we apply a therapy, it's necessary to find a good biomarker so that we are giving the correct therapy to the correct people, especially when you see some of the adverse events. Okay, so this is just a quick screen capture to show that it's very relevant. It's there, it's on front page of all the major reviews. Three main types, essentially people looking at cancer vaccines. So the main thing of immunotherapy is essentially if you Google it and get kind of Wikipedia, it's essentially the use of the body's immune system to fight against disease. Okay, so with that in mind, what people are trying to do is they're trying to leverage the power of the patient's immune system to fight against or battle against. I'm not sure what words to use now. But essentially to fight specifically against the tumour. So vaccines are a way where people might provide a kind of foreign agent that would be injected or delivered into the tumour mass, and that would invoke an immune response on the tumour. That's kind of one approach. Cell therapies, I'll kind of touch on a specific case study later in the slides. And also a very popular one at the moment is immune checkpoint inhibitors. So these are basically molecules within the immunity cycle, which are used to actually control the immune system and make sure that we don't have too much of an immune response. So where you start to get autoimmunity, things like rheumatoid arthritis and things like that, examples of autoimmunity. But when we do find that the tumour is present, sometimes we're looking to block those control mechanisms to give the immune system an extra boost against the tumour. And so I think the previous speaker was Professor Complicated. You now have Mr. Simple, okay? So we're just going to walk through it, start at the beginning. So with regards to cell therapies, essentially introducing the cell, we're all made up of cells, so I've tried to pitch the presentation with a view to there being lay people in the audience, and not everyone is a Professor Complicated, me included, far from it. But essentially, it's a kind of basic building block of the body, okay? And people are talking about nucleic acid, they're talking about DNA, RNA, and proteins, all present within these cells. And all these cells, you know, come together, and they're forming tissues. The tissues are then forming an organ inside the body, and then we get groups of organs that together provide a biological system. So you can think of like the digestive system involves a number of different organs and as a system it has a particular function. The immune system is the same. It has various organs. There's a huge component in the blood, the white blood cells running around the body, and it's specifically some of these white blood cells that we're looking to leverage with these cell therapies. So these are the different cells of the immune system. So quite nicely named so that you can put big letters on them, fortunately. Essentially, what we're most interested in is these cytotoxic T cells. They're essentially the cells that are going and killing, it can be viruses or tumor cells or various things in the body. Okay, but the problem with trying to leverage the immune system is that it's really, really complicated because there's so many different cell types. All the cells have various functions, and the controls of those functions are through different chemicals like chemokines and cytokines, and there's various signaling pathways. So it's a very, very complex network, and everything is interacting with everything else. It's a bit like the tumor microenvironment that was shown in the previous presentation. There's tumor cells, there's normal cells, there's some immune cells that have seeded themselves trying to act against the tumor. And so it's really a complex network of cells, signals going on. And so that situation, that tumor and that microenvironment is different with each tumor in each patient, or if there's metastasis, then basically, you know, the metastasis will look different from the primary tumor, and so it's really necessary to be able to pull apart what the different scenarios are looking like in order to be able to treat them correctly. Okay. So one of the major cell therapies that's being used and is massively being developed across a lot of the labs that I'm visiting is the use of CAR T cells. So there's a critical part on each of the T cells that pretty much defines them, and that's the T cell receptor. And this is the part that recognizes the foreign body that's invading the patient. So you can almost define that you have a population of T cells, and you will have some with this T cell receptor, some with this, and you can measure kind of like the diversity of your T cell population. And then if there's a specific virus or a tumor cell that's in high abundance, then you will get, hopefully what you're trying to achieve is a T cell that is reflecting that tumor cell. It can bind to it and it can kill it. And that's essentially what you're trying to mimic. So the actual, the CAR T is actually where you're taking a patient's own T cells out and you're creating, sometimes you are creating a specific T cell receptor. Sometimes you're just trying to expand the diversity of the T cell receptors, but essentially you're genetically engineering that specific part of the T cell. Okay, once you have either your specific T cell receptor that will target that person's tumor, or you have your increased diversity of T cell receptors, then you would expand and grow more of them in the lab. And then at that point, you would essentially provide this basic drug, which is essentially like a living drug that's infused back into the patient further down the line. So it's essentially taking the T cells, trying to change them so that they're specific to that disease, and then expanding the number of them, and then reintroducing them to the patient. And then you're waiting to see that those T cells start to clear the tumor cells. So there is kind of technical part to it, and it is necessary. You know, sometimes you will kind of blindly almost try and just increase the diversity of the T cell receptors and try and increase or invoke an immune response, or you can take specific known epitopes and create T cell receptor, knowing that it will actually react against the tumor. So it can be specific or it can be increased diversity. So this is just an idea of the kind of overall workflow. There's a number of steps. It's quite complex. It's also very expensive as well. So although it's shown very good results, there is a need to be able to speed up the process and also to make it more scalable, because it's quite a kind of customized kind of creation of it's a very personalized medicine, because it's essentially your own white blood cells. So we're just trying to ensure that we can optimize this process so that it can be manufactured on a larger scale. But the idea is that, you know, in doing so, you're introducing these T cells to your body. And one of the great things about the immune system is that it has a memory function. And so this is lending itself to a good, what we call durable clinical response. So the idea is that it's not just, you know, clearing the cancer now, but then it's given the immune system the memory, so that if that is returning, that same tumor, then the T cells are ready to be released again. And so recurrence of the same tumor should be restricted significantly. So there's, along with the, we said at the beginning I'm a molecular biologist, okay? So the cells are super interesting, but I work more in the biomarkers, and so looking at things like the levels of mutations that each tumor has, so how much is it going to respond to an immunotherapy? And we actually now have the ability through molecular biology to actually characterize down to kind of like nucleic acid base level the different T cell receptors. So we're able to actually give a specific sequence of the part of the T cell that's binding. Also importantly, we can give the frequency and the rank order of the population of T cells. So the idea is that we're actually using these T cells as a biomarker themselves. So the idea is that you could take kind of a blood draw, and you basically analyze all the T cells in the patient, and you can check how diverse they are and how many different types, how many, we call them clonotypes, how many different clonotypes of T cell receptors there are. And then you can report that back. Then you would perform your cell therapy. And if you've created a specific CAR T cell, then you can track that, because that specific CAR T cell receptor sequence will be in here, and you can say, I can see it. I can see the frequency it's virtually taking up like most of the T cell population, and you can monitor that over time. And so rather than just giving the T cell therapy and sitting there with the fingers crossed, you can actually physically start saying, okay, I know that the T cells are happening, are working. Likewise, if you just provide diversity to the T cell receptor and then introduce it, then you can all of a sudden, you can see if it goes from a very even population of diverse receptor sequences, all of a sudden one clonotype increases massively, then it's a reflection that there's a corresponding tumor progression, because you can start to see this reflected in the T cells. So you really want to see, do I see my introduced T cell receptor, and is it expanding? And this is just a complicated picture of what's actually happening. Interestingly, there's also really relevant information in these three regions here, CDR 1 and 2, particularly have some germline variation in them. So some heterogeneity between people that's inherited. And this is thought to be linked to the filtering or non-filtering of autoimmunity T cell receptors. So there's a thought that there's some links in variants here that are giving us more information on adverse events. So that's also something we're looking into. So I'm just going to end with a case study. So basically, it's kind of the most famous cell therapy case study, I think. So just to give it some background, we have approximately 62,000 new cases of leukemia each year in the US. Mostly they're treated successfully with chemotherapy, mostly. But for the people that it either doesn't work, or there's also a group of people that start off sensitive to the chemotherapy, but then they become resistant to the chemotherapy and the disease progresses. There's not really a lot of other things that people can do. And what we actually had was FDA approval on April 30th, 2017 of the first CAR T-cell therapy for leukemia in children. And that was based off a specific case. A little girl, certainly a little girl at the time, Emily Whitehead, in 2012, she was really poorly. After initial response to chemotherapy, the disease progressed, and essentially they performed a CAR T-cell therapy, and you can see, without getting too technical, with the infusion of the CAR T-cells, there's a massive drop in the tumor load, and basically complete clearance of the cancer cells. And this is happily, this is Emily, 5 years cancer-free in 2017. So that's everything from me. So thanks a lot for your attention.